so hard in Geneseo, but all of what you see is essentially field work done as part of two classes, one at SUNY Brockport and one at Geneseo. Okay, so let's start. We'll let the important contributors do the talking. So I'm going to ask the first Geneseo group to come up, and all I'm going to do is read the title, and they will tell you the rest. So the title of this presentation is Size Distribution and Community Filtration Rates of Cladostrum Zooplankton in Several Lakes Potentially Influenced by Alewife Predation. All right.
Hi everyone, I'm Samantha.
I'm Steve. I'm Michael.
I'm Emma.
And as Dr. Bosch said, we studied the range of body size and filtration rate for multiple species of tiny crustaceans in 5 nearby lakes with a focus on how body size may be influenced by the presence of alewife in those lakes. And the lakes that we looked at were Canisius, Hemlock, Seneca, and Keuka lakes, with Loon Lake being our control lake, which did not have any known populations of alewife. So to give you guys a little bit of background, the tiny crustaceans we looked at eat microscopic pieces of algae, and a bigger animal is able to eat more pieces of algae as well as larger sized pieces. And alewife are an invasive species of fish who like to eat larger sized crustaceans. The results of our study showed that Loon Lake, which does not have any known populations of alewife, had crustacea that were nearly 3 times the size as the other lakes, which did have alewife in them. And to take things a step further, we performed a filtration rate calculation, which takes into account body size and species abundance. And that was measured in % of a meter cube that was able to be filtered a day. And that kind of gave us a way to look at how much these crustaceans are able to eat and how that can impact the lakes that they're in. So the results of this calculation showed that Loon Lake had a filtration rate which was just under 26%, and Canisius and Hemlock had filtration rates of 7% and 3% respectively, which is obviously much lower. And the importance of the study shows that these tiny crustaceans have a huge impact on their lake communities, and alewife populated lakes have much smaller average body size for crustacea, and they have a lower filtration potential, which means that less algae are able to be consumed. And this could be a major factor in overall water clarity and water quality, and is something that should be looked at to improve these factors in our nearby lakes.
All right. I have to be careful because some of these students want to take my job. Great job. Okay. The next group presenting is also a Geneseo group. You'll see the first 3 are Geneseo students in my class. And this title of this project is their unique community of little zooplankton, or Cladostrum in this case, little crustaceans, in the nearshore regions of Canisius Lake.
So as Dr. Bosch was saying, we decided to look at the difference in communities between the nearshore and offshore. I'm Madison.
I'm Krista.
I'm Alex. I'm Raul.
So the increase in alewife in the offshore region of Canisius Lake has led to a decrease in the crustaceans in the offshore community. So we decided that it would be important to focus on the nearshore community and see if there's a difference between the two there.
Yeah, so how we went about this was we took offshore samples from the deeper waters around 7 meters all the way up, and then nearshore samples, and plated those and compared the Cladostrum in size and abundance.
So we had a few key species that we were looking at. The first was the Diapenosoma, which through lots of studies on the offshore greatly decreased around from 1985 onward. And then what we knew as the Cereodaphne species 2, which ended up being the Cymocephalus and the Chirodus rotundus, were all species that were more abundant in nearshore populations than offshore populations. So what we wanted to do is compare the 2 communities to each other. So we started by looking at a community similarity index to check out the differences in percentages between the 2 communities, and then we transitioned to a size distribution comparison graph.
And we found that the most abundant species, they're found in both the nearshore and the offshore communities, while the more rare species like the Diaphanosoma, the Camphocereus, the Cadiaceus, and the Cymocephalus were found in more in the offshore than the nearshore. And these four species were generally larger in the nearshore than offshore.
Yeah, so we were thinking that this might be due to the alewife being present, causing there to be a refuge for these little crustaceans towards the near shore.
Okay. This is going to be the final poster from the Geneseo group. And whereas these first two have looked at the crustacean zooplankton community, this third group was interested in looking more closely at cyanobacteria, or blue-green algae as you might know them. So the title of their talk or their poster is Sequential Dominance of Cyanobacteria and Diatoms in the Late Season Phytoplankton Community of Leech Lake.
I'm Abby.
I'm Steve. I'm Nathan.
And I'm Isabella.
We looked at the changing dominance of the blue-green algae and the diatoms in Geneseo Lake from the late summer into the fall season. Cyanobacteria like the warmer temperature of the water and are toxic to the ecosystem, while the diatoms are thrive more in the colder water and are beneficial to the ecosystem. We saw in previous studies that the cyanobacteria, the blue-green algae, are persisting longer into the late summer. So we wanted to see if the diatoms were being replaced by the cyanobacteria or if they're still coming back in the fall.
So we went out onto the lake and we took 4 samples from early and late September and early and late October. And we brought those samples into the lab, put them under a microscope, and we individually counted filament cells and individual colonies that we were interested in.
And as you can see in our pie charts, our results were generally what we expected, with the highest cyanobacterial abundance in our early September sampling date and our highest diatom abundance in our late October sampling date. This was consistent with historical studies we had looked at as well. And this is extremely important to the lake because cyanobacteria are toxic and diatoms are an important food source for fish and zooplankton preparing to overwinter in the lake. And maintaining this population of diatoms is integral to maintaining a healthy lake in the future.
All right, so now we're going to shift gears and some of my Brockport students are going to talk about their work. So most of the Geneseo students working on Geneseo Lake. My students had probably more diverse, I guess, ecosystems that we were focusing on. So the first poster is by Jacob Cellino. He's going to be talking about his work with microplastics and also harmful algal blooms.
Question. Arthropods, not. So like he said, I'm Jacob, and I looked a little bit different from what other people did. I did a bit of a review on what's going on with literature. So arthropods are common benthic macroinvertebrate that live in the bottom substrate. They make up a major food source for a lot of fishes, and they also important shredders in the ecosystem. And then microplastics, as a lot of people may know, very small plastic particles, is sort of an emerging contaminant. And there's a little bit of research on them, but not as much in freshwater ecosystems. So what I did is I looked through literature with using terms that related to these things. Then I looked through the literature and found results that applied in the situation. I found 18 papers that reviewed microplastics and arthropods in freshwater systems. And they were pretty evenly split on type of assessment they did between chronic assessment and acute assessment, though most studies were lab studies. The primary particles that they looked at were polystyrene particles, which float on the surface water, so that's really not super applicable to them a lot of the time. And then ultimately, a lot of these papers concluded that there needs to be more research done. There isn't really a ton out there. And a lot of these studies only focus on lab conditions and direct contact with these plastic particles, whereas in nature, they're not just in lab conditions. So a lot more studies should be focused on field interactions and particles that have been weathered by the environment, and also particles that have come into contact with various toxins in the environment. Thank you.
So next up, we have Braden, who's going to be talking about nutrient dynamics and internal loading of phosphorus in Loon Lake, New York.
So, as you said, I'm Braden. Over the summer, I received a grant through SUNY Brockport called the Summer Undergraduate Research Program, and that allowed me to work on Loon Lake. At Loon Lake, we do have a historical deep site that has been sampled and monitored for about 3 years now. So I was pretty much just continuing that research. And I was also focusing on nutrients that are key components for algal blooms, mainly things like phosphorus. The thing with phosphorus is that without it, obviously algal blooms can't really occur. So as the lake stratifies over the summer, the sediments, since they're in a low oxygen area, they do release quite a bit of phosphorus. So what we were seeing is that the lake stratified earlier this summer than in past summers. And as a result of that, we actually saw a huge increase of phosphorus sitting at the bottom of the lake. So the amounts we were seeing about July of this year, we didn't see last year until about October. So quite a bit. And at the surface of the lake, well, compared to from the surface of the lake to the bottom of the lake, we actually saw an increase of 1,900% of phosphorus. So quite a bit. And what that means is that when the phosphorus sits at the bottom, the algal blooms the algae can't really get to it to cause a bloom. But when there's a huge mixing event, such as windstorms that we had in the area, that causes phosphorus to rise to the surface and become available, which actually happened over the summer at Loon Lake. So we did see a huge bloom pretty late in the summer. And the issue with that is that some algal blooms are toxic. They do produce cyanobacteria, which obviously if the lake stratifies earlier in the summer, there's more phosphorus in the water, then we do see there's probably a likelihood of more of these toxic algal blooms. So more research is needed to see if this increase of phosphorus is an actual trend, or if it's just basically just like it was just a weird hot summer that caused this increase.
Now we're going to shift gears to a different ecosystem, but also talking about productivity and trophic state of this ecosystem. So next group is going to be talking about impacts of the Braddock Bay Barrier Reef Restoration Project on water quality.
I'm Liam, Braden, Caleb, Jacob, Shane. So yeah, this second, just like I said, we were talking about the impacts of the Braddock Bay Barrier Restoration Project. And Braddock Bay is an embayment located southern coast of Lake Ontario, approximately like 11 miles northwest of Rochester. And over the last 100 years, about 106 acres of wetlands in the barrier have been washed away through wave-driven erosion. In 2016, the U.S. Army Corps of Engineers completed a barrier restoration project and began sampling before that and after that to compare the trophic state and water quality.
Water samples were collected at Braddock Bay before, during construction and post-construction to compare the trophic state index of Braddock Bay overall. And as you see in this figure, they broke up the segments of protected to non-protected areas of Braddock Bay to see if they differed.
So when looking at the results, so other than the trophic state index, which can be calculated in a couple different ways, we took an individual look at the individual nutrients. For example, nitrogen, phosphorus, TSS, chlorophyll. So by running multiple different tests, we could see that there was actually no significant difference between before and after the barrier breach was built. But with a closer dive, you can see that starts to be a trend. It is not significantly enough to say there has been a difference, but there's a trend, which can show that there needs to be more data presented. If you can see a quick graph of the TSS, there has been a significant decrease from when it was first started in 2015 until about 2020, and then 2019 to 2020. And then for about 2 years, it did start to increase, but after looking at the decrease, 2 years in the grand scheme of things is not a lot of time. So more data is definitely needed to confirm our results that there was no significant difference between before and after the individual nutrients.
Yeah, and we calculated Carlson's TSI based on chlorophyll A for all 7 sites to compare pre and post construction. And we found there was a significant difference based on a t-test between pre and post construction. And
was it higher or lower?
it was lower. I don't know if you can see this figure here or not, or if it's, yeah, okay. Yeah, it decreased. That's good. Yeah. Yeah, so although significant differences were found, the bay remains eutrophic. And this is concurrent with the goals of the project, as they didn't want to necessarily change the water chemistry of the bay, rather maintain it.
So the project plans to slow erosion by 55% and save around 27 acres over the next 50 years. And we found that the total phosphorus concentrations remain similar over the years sampled. And to reduce the total amount of phosphorus, the effort will be needed to reduce external loading from the phosphorus-rich tributaries.
All right, so now we're going to shift gears back to Loon Lake and talk about aeration, one of the potential ways we can deal with harmful algal bloom. So the next group is going to talk about the effects of aeration on physicochemical properties of Loon Lake.
So yeah, we also looked at Loon Lake, but we looked to see what the effects of aeration would have on the properties of the lake. We looked at dissolved oxygen and also chlorophyll concentration, and we had 3 main hypotheses with the data we were given. We hypothesized that chlorophyll concentration would be reduced at aerated sites. We hypothesized that dissolved oxygen levels would increase at aerated sites, and we also hypothesized that these changes would be amplified the longer that the aeration was continued.
Okay, so the aerators in Loon Lake were installed early 2021, and we categorized them into 4 main treatments. These are aeration near shore, aeration control, offshore control, and offshore aeration. So at each of these sites in both 2021 and 2022, we sampled the physiochemical properties about 1 to 2 times a month from May to September. And with these, we collected data such as temperature, pH, dissolved oxygen, chlorophyll. And using this data, we calculated the trophic states at each site. And we created a few figures here that Nicole will tell you about.
So unfortunately, none of our hypotheses were proven to be accurate. We expected chlorophyll to decrease in the aerated sites, but it actually spiked near the end of the sampling period. We expected dissolved oxygen to increase at the aerated sites, but only 1 site was actually statistically significant. We calculated the trophic state in the indices for May 2022 and August 2022. They didn't change that much. They all stayed in the 40s, which means that they're still moderately productive. Okay, so overall, our results were not what we were expecting. This was such a short-term study. We suggest that possibly we keep studying and keep seeing what happens over time. It seems like aeration is more so a short-term solution, more so than a long-term solution to helping decrease the excess nutrients that are in Loon Lake. Loon Lake, I'm not sure if anyone's familiar, but it has a lot of agriculture around it. So the runoff might be, the reason why there's so much excess nutrients. So it definitely is something that we should keep studying. Yeah.
All right, so now we're going to come back to Canisius Lake, and next we're going to talk about hypoxia, anoxia, and also trophic state of Canisius Lake over time.
Christara,
I'm Kevin. James. Colin. Thomas.
Christine.
So we looked at hypoxia and anoxia of Canisius Lake over time from 1910 to 2022. Hypoxia or low dissolved oxygen levels is mainly attributed to harmful algal blooms of eutrophic lakes. When the algae dies, it sinks to the bottom and decomposes by a process through cellular respiration, which causes the hypolimnin of the lakes to become hypoxic. We study Canisius Lake, which supplies drinking water to Geneseo and Abba.
So for our methods, obviously we're not immortal beings. We can only collect data for so long. So for our class period in this fall, we collected data for Canisius Lake, and we also had legacy data, not consistently from 1910 to our collection date, but from 1910, 72, 73, 78, and 96. So there's a big gap in the 1930s and 40s for some reason. We also calculated the TSI and all these figures using Microsoft Excel. And we also did a regression on the TSIs to find out if there was a causal relationship between TSI and hypoxia and anoxia.
Yeah, so there were 9 reports that we accessed. 4 of them were from the 1970s and prior, and 5 of them were from the 80s and more recent than that. So we kind of just split that down the middle and did a t-test and found significantly higher trophic states since the 1980s compared to before the 1970s. But there was no significant difference in dissolved oxygen at the surface or the depth at which it became hypoxic. And we had regressions to test the relationship between dissolved oxygen parameters over time, as well as TSI over time, and also the relationship between TSI and those dissolved oxygen parameters. And none of those were significant, which wasn't what we were expecting. And so specifically, if you look at, well, most people probably can't see the figure, but the trend in trophic state over time, there's kind of a jump here in the 70s, and then the trend has been a little bit reversed, where it's been decreasing more recently. And so that's, I think, a lot of why we didn't see significance there, because it's starting to gradually decrease where it's been increasing before. More.
So there was an implementation of various management practices beginning in this 1970s, which likely slowed down the process of eutrophication within the lake. The relationship between trophic state index and surface dissolved oxygen was negative but noticeable, but not quite significant. We only have 1 data point per report, so it may not have been a strong regression. Canisius Lake is also widely accepted to be eutrophic. However, our data shows that it's mesotrophic due to the TSI value being between 40 and 50. There's been recent improvement in dissolved oxygen, but the trophic state still has a way to go to reach 1910 levels. And then, as Kevin said, there's figure 4, it shows the decrease, showing that it might be heading in a positive way.
Last but not least, we'll be staying at home near the Geneseo River. And the last group is going to talk about restoration or recovery practices for lake sturgeon in the Geneseo.
Everyone wants lake sturgeon. So we kind of did a secondary analysis of previous data that was already acquired by the DEC, New York State. So they did population analyses from, way back when, kind of like they said, there's a gap in some of the data, so it's, as accurate as it's gonna be. But we made, what's up? Yeah, so lake sturgeon have been declining in their numbers since the 1900s due to overharvesting and overfishing and all that. So that allowed for New York State to kind of add these implementations and plans to restock the fish. So that's kind of where our data comes into play. So we made a population assessment of our own using all the data that was provided from the DEC and other sources and kind of put it into a table here. And you can see there's a variety of numbers we're getting in each of the 7 locations, and that could be due to different sampling methods that they used. Some were used by pit tagging or mark recapture analysis or observational counting. So there's some variety in our data. But yeah.
So we created a timeline of the historical abundance and the decline in the increase of sturgeons. And so our first information that we got was more than 15 million lake sturgeons were found in the New York waters. And that's like in this picture here. And then after the decline of like habitat alterations and over harvesting, their populations just started declining where they got to 1% of their native range, which is over on this side. And then we created stocking programs in 2006. We stocked over 44,000 total sturgeons. So it shows back on that photo, it's like before and after. And so the numbers really increased after that.
The results, they show that the efforts led to like self-sustaining populations. They've seen an increase through the monitoring and research, but there still is like a lack of spawning locations and like the locations of where they're from or like where they go to spawn, like the status of the habitats.
Yeah, so further research needs to be done with the egg mass, like they were counting the egg mats that they found. So they need more research to figure out the accuracy of those numbers. But this table shows locations met the goals for the egg mat numbers and adult population status as well. So you can see some of them met both, some of them met one, some need a little work, but yeah.
So now we're gonna move on to phase 2, right? I'm gonna take this podium on the road. I'm gonna go in order around here. Everyone else, if you want to think of it like a trade show, just go talk with the various teams. I'm going to be hosting the Zoomers that are queued up here. And we'll see if we can get some questions from them for you folks and share with them what you have been working on. Thank you. I will be using this as a microphone. So if I hand this to you, it means you're talking this. I didn't know they could do this. This is really cool. So we could isolate this conversation from all the others that are going on simultaneously. All right.
Yeah, so the poster presenters, you don't need to have everyone by your poster at all times. It's important to maybe have 1 or 2 people, but also the opportunity to go around.
Hello there, I hope you can hear me. At this point, we're going to move around to the various screening sessions. If you've downloaded the PDF file, I'll be going in the order of the PDF file, except the last 3, actually. The last 3 will be going in the reverse order. I've got a little mix up there. So if you want to follow along, look at your data on your file, that would be great. So let me move us. What's this section? Excuse me. Let me see through here. So this session is the size distribution and community filtration rates of some kind of zooplankton in several lakes, potentially influenced by alewives. And it might help you as observing the Zoom session here to pin my screen to the front so you can see what we're seeing. I'm going to do that right now. Here we are. Hi guys. Nicely presented. One of the interesting things I noticed about the SUNY Geneseo presentations is they were all biology related. And if you notice the SUNY Brockport presentations were environmental, a lot more physical stuff. I was very impressed with that. I'm going to try and introduce and allow for questions from anybody on the Zoom session. I think I have to turn the speaker on to make that happen. Charlie, can you hear me? I can. Loud. Can you hear me, Charlie? I can hear you, Carl. I have trouble hearing you, so I'm not sure. Maybe chat would be the best way to submit questions. So I found this a very interesting study because you found different size crustaceans. I can't hear you. You probably need to hold this close. Oh, hold it real close. Okay. We found different size crustaceans in lakes that had been infested by alewives. That's the fundamental difference. I wondered when I was reading through your paper, why Loon Lake would have such a broad spectrum as well as larger. The others are very narrow, with the exception of Seneca. Any idea on why that is the case?
Maybe it's because of the pressure of the alewife. It just causes that diversity to be limited into a specific niche in the ecosystem. And that size is just smaller on average. And I think since it's a smaller on average range, it doesn't give them so much range for diversity and size to be smaller or larger than what Loon Lake is, essentially.
So that implies the alewife has a limited size or a limitation on that size of zooplankton. So it can't eat that size zooplankton.
They only eat potassium that are about 0.8 millimeters in length. 0.8 millimeters are large.
Do we get any questions from the Zoomers? Yeah. Let me go off to the side.
For those on Zoom, you can hit the space bar.
I'm sorry, Charlie. Say that one more time.
For those on Zoom, they can hit the space bar and ask for questions.
Can you ask him to type in?
I'm sorry, Charlie. It's noisy in here, and this is not working as well as I had hoped. Perhaps you can type your question in chat. Is that okay? Because I can't hear it. I had a couple other questions, so let me ask other questions. how did you determine the volume of water that you were filtering through?
So we took a net from 7 meters down and we knew the circumference of it and dragged it up, and as it filtered the water, so we basically calculated the volume of like the column of water that we pulled it up through, and then we were able to use that to determine the abundance, basically like extrapolating how much there was in a milliliter of our sample to how much there was in a milliliter of the actual lake.
So that assumes that 100% of the water in the column goes through the filter. Is that a reasonable assumption, or wouldn't some resistance cause some to spill out?
Probably the net would cause some of the water to spill out, and it wouldn't be a perfect calculation, but it's as close as we could get.
Fair enough. I'm just curious about that. And several times I read about filtration rates, and I was a little confused by that term. Can somebody give me a layman's explanation of what filtration rates are?
So filtration rate was essentially looking at how much each of these tiny crustaceans as like a species community could filter in the lake. So these crustaceans eat algae. So we looked at the body size of each of them and we used our abundance data and we plugged it into this calculation and it spit out a number of how much of 1 meter cubed of water in the lake could be filtered, or essentially how much of that algae could be eaten by the community of these crustaceans in a day of a meter cubed. Yeah.
So it basically tells you the concentration of filtering in the sense.
Yeah.
Okay. Let me see if I can read this. Oh, we're trying. It's hard. I think that's my question. I don't see any other questions on chat. I don't see any questions on chat, so thank you very much. Thank you. Excellent paper. I really enjoyed reading.
So alewife obviously is not a good species to have in these lakes, right? So what do we do about it?
Well, I don't think you can just remove them because they're an invasive species, and anytime you have an invasive species in for a long time, removing them would affect the whole ecosystem. What?
Trying to get us to say why.
That's not a good idea. That's a bad idea.
That's a bad idea.
You don't introduce more things because it'll just throw the ecosystem out of whack even more.
Thank you, guys. Well done. We're going to move on to the next paper, the next poster. I muted that. That's no problem. Charlie, can you hear me now?
I can.
This is much better. I think the microphone system was not muted. And that made it difficult to hear because it was picking up all the conversations in the room and putting them over Zoom. I apologize for that.
No problem.
Much better. Much better. We're here at the second poster. For those of you that are following along on your PDF file, this is the second one from the top. And they were discussing the unique community of zooplankton in the near shore regions of Canisius Lake. So the littoral section, all the area near shore compared to the deep water. And what did you learn?
I'll take it. I didn't know what I was expecting when we started this thing because I wasn't sure what the difference would be because I didn't know too much about the alewife besides the fact that they were invasive species and there were a lot of them in the lake. But through researching, we learned that alewives mainly stayed offshore and rarely would predate on the nearshore populations. So we started to think that maybe that there would be differences, like a lot of the zooplankton and philosopher that were offshore that were being eaten by the alewife would move near shore for like refuge under the macrophytes. And I was actually kind of shocked to see there was actually a pretty big difference, only a 50% similarity between the two communities.
I noticed a similarity between this paper and this project and the one behind the first one. And what it seems like is the abundance near shore is less affected by the alewives, yes? Which parallels what happened in the first paper when they looked at Loon Lake, which didn't have alewives, right? So my question to them was, why, in addition to having bigger samples, do we have more? Why is there a wider range?
I think that the biggest reason that there was a wider range is because we think that there was the nearshore was a refuge for these Colostra. So since there's less alewife nearshore, we believe that that's why there's a larger variety of them.
So in a way, these different variety of species could escape the fish that are eating them. That's about it.
Yeah, fair enough. So of course there's more because they're not being predated.
Pretty much.
When I looked at this graph, I wondered why so many sizes were at that one level. Did anybody look at the data, understand why that is?
You could take it. I can take it. I did the graph. I think it had a lot to do with the amounts of the different sizes that we had. A lot of them seem to be like, if looking at couple of pictures, a lot of them looked around the same size. There are minuscule differences between the two. And so the big spike on the nearshore and on the offshore as well were all like the Bosnino. They were all around the same size. But it seemed like a lot of the other ones varied slightly, like they were around the same size, but very different, like slightly different. And so that caused a lot of them to end up being the same amount, but like different.
Oh. I'm not sure I understand that yet, but I'll have to think more about that. And I noticed on this graph, the scale here is different. So it's only 3 years between these 2 points and 5 years between the next 2 points. That's just because of availability of data?
Yep, it's what we had.
So we need more data.
Pretty much.
Okay. Anything else you'd like to contribute?
Yeah.
Charlie's right. Sorry, Charlie. Thank you for the suggestion. I'm a pain, but not a blast, right? My mom always said. Anything else you'd like to add to the presentation? I really enjoyed this presentation. The biology focus of SUNY Geneseo is evident when you look at these, as opposed to the environmental focus, which is also very interesting, of SUNY Brockport. Thank you.
Thank you.
We'll move on to the third one. I'll try and remember not to stand in front of the camera. Probably about as good as we're gonna get. So here we're at the third poster. The one with the beautiful rainbow in the upper left-hand corner. Sequential dominance of cyanobacteria and diatoms in late season phytoplankton community on Geneseo Lake. I had a question related to this third paragraph here. And my question is there any follow-up on this statement here? The goal of the 2022 study was to describe the September to October phytoplankton community in Geneseo Lake and determine if the fall dominance of diatoms had diminished in this age of cyanobacteria. I wondered if there was a follow-up. That sort of implies, just talking about 2022, should be looking at this, maybe not you as a group, but it will be looked at in the future. Is there any plans for that? Do you know?
So basically, what we found is that the diatoms are continuing to dominate in the fall, despite an increased cyanobacteria prevalence early in September. So luckily, even though cyanobacteria have become more dominant earlier in the year, diatoms are still persisting.
Good. Thank you. I also appreciate the fact that you use a lot of temperature data.
Yeah.
Very good. Once again, this is a biology, very much a biology paper as opposed to environmental paper. And it really struck me the difference in the two courses. Are you folks all biology majors? Is that
Yeah.
Yeah, good. Nice. Well, Dr. Bosch is a neighbor of mine and he does a great job. Anything that you particularly learned in this project that you haven't said you'd like to share?
We learned a lot of, excuse me. We just learned a lot of types of diatoms and phytoplankton through looking through the microscope, but yeah, all of our findings are up here, so.
When I hear the word diatom, it makes me think diatom because it's kind of spelled diatom. Does that have anything to do with atoms? What's the deal?
Not to my knowledge. I don't know. I don't think there's any connection, at least as far as I know. But that might be a question for Bosch. He might know more about that. Yeah.
Thanks, guys. Nicely done.
Thank you.
Nicely done. Move on to our next one, if you don't mind. We're now moving into the reign of Dr. Michael Chislock. And this is the paper or the poster on microplastics, which I was surprised to learn are bigger than I thought. When I think of micro anything, I think really small, but you had a 5 millimeters, half a centimeter. That's a fairly big, still pretty big piece of plastic to be floating around. So I was surprised at that. Was that a large portion of the?
No, most of them actually focused on plastics closer. Most of them focused on plastics closer to the 100 micrometer range. Sorry, and the 100 micrometer range, which isn't super representative of what you find out in nature. So that's another problem. But 5 millimeters is typically the upper end of what they consider microplastic.
And I noticed these terms, which I'm not familiar with, Palearctic and Nearctic. I had two questions for you on this. One is, it seems like Nearctic is only a very small part. And what do those things mean?
Okay, yeah. So Palearctic is, well, these are biogeographic regions. Palearctic is the old Arctic region. So it's the northern hemisphere in the old continent. So Europe and Western Asia is typically where they consider Palearctic, and Nearctic is, broadly speaking, just North America, northern part of North America. So it's just biogeographic regions where these organisms are found. So, as you can see, most of them focus on the Palearctic, which we're in European countries typically, which is not as good for us because we have the Nearctic species, which only a couple of studies focused on. That's mainly the big difference between them.
There's a big difference between the Palearctic and the Nearctic, or are they fairly similar?
So like I said, they're two different regions of Earth, so they have different, they have a little bit different ecosystems. They have different organisms that dominate those ecosystems. And like I said, I'm more interested in the near Arctic, which is where we live. I think everybody here is probably more interested there because that's where we live. But a lot of the research is kind of coming out of Europe, so that's why a lot of these are the pale Arctic species.
Great, thank you. I noticed you were focusing on polystyrene here as a dominant plastic particle. And I would say it's clearly the largest. So I guess that makes it the dominant, but it's not by any means the majority. It's one of many, right?
Yeah, so there's a lot of different plastic types, and a lot of these studies focus on polystyrene, even though that's not necessarily the most dominant plastic in the environment. Typically, what's considered the most dominant plastic in the environment is polyethylene, which comes in two primary forms, from low density polyethylene and it's in the other category. It's high density polyethylene. Very few studies actually focused on it. Those typically are considered the most common forms of polyethylene. And why the research is focused on polystyrene is not super clear to me. Oftentimes polystyrene, you can think of it as like packing peanuts. It floats on the surface, so it's not super applicable to these benthic dwelling organisms. So that's why it's a little bit concerned that a lot of studies did focus on the polystyrene.
Well, thank you. I wanted to make sure I asked if there was any questions from the Zoom community before I moved on. I don't see any in the chat. I don't hear any, so I guess it's not. I think this is a very interesting paper. I really like poster. I really like the fact that you use the work of others and you sort of correlated that for us. And found 11 papers. We found neutral population.
Yes.
Or individual response to plastic, which is sort of encouraging.
Yeah.
Unfortunately, 8 found negative response, which is a little bit more discouraging.
Yeah.
But it is interesting to see the collection of literature summarized for us. Thank you very much. Excellent. We are here with Braden Link. Hi, Braden. And Braden did some work understanding nutrient dynamics and interloading of phosphorus in Loon Lake. Now, does Loon Lake have an outlet?
No, so that's the thing with Loon Lake. There's actually, I'm not sure if you can see that here. Thank you. There's actually only 2 streams that come into Loon Lake, and most of the summer those streams are dry. So Loon Lake is fed by an underwater spring pretty much, and so there's pretty much no outlet for it. But the level of lake does focus, it depends on the spring that's feeding it. But the thing is that since spring's underground, we don't really know where it's at or what's all coming in with it.
Let me see. Question for you. Yeah, I was reading. You said. Let me go to this side. So you said here in the last few samplings, water samples from the last few depths had a sulfur smell. Can you tell us what that implies?
That implies that the bottom of the lake is deprived of oxygen, which can actually be seen here in this this graph. And basically, when the bottom of the lake is deprived of oxygen, the sediments release phosphorus, and it's the phosphorus that gives off that rot egg sulfur smell. So in the last few samplings, the water had like a gray tone to it, and it smelled very badly of phosphorus and of the sulfur. So that just means that, as we can see in this total phosphorus graph, phosphorus was just an extreme amount at the bottom. So the stronger the sulfur smell, the more phosphorus is in the water.
Or maybe the more phosphorus in the water, the stronger the sulfur.
Yes.
Very good, thank you. I also was interested in your comment here about more data being necessary to indicate if this is indicative of a long-term trend or earlier onset, so summer stratification. Do you have any sources potentially for that kind of data could be coming?
I haven't really looked into a lot of literature, but the issue, we do have, like I said in my intro, 3 years of data that's been collected at this one historical deep site. So comparing the data that we collected this year to last year and the years before that, this year the phosphorus and the lake stratifying a lot earlier, and the huge amount of phosphorus. We don't know if it's an actual trend or not. This year is also extremely hot, which causes the lake stratify earlier and release phosphorus earlier. So it's going to probably have another 2 or 3 years of data collecting to see if it's an actual trend or if it's just an extremely hot year and it's there. Yeah.
Interesting. Thank you. I think it's a very interesting lake to study because it's so small and well contained.
Yes.
I noticed I'm continually struck by the environmental focus of the Brockport papers versus the biology focus of the Geneseo papers. Just very interesting because they're 2 different courses, I think.
Yes, this is technology. So it's a steady.
Right.
So yeah, it's definitely more water-based, like nutrient-based. Technology is more like water quality, like nutrient-based. We do talk a little bit about the organisms that live in it, but not as in-depth as Geneseo does. I'm not sure what their course is. But yeah, so with technology, we just kind of do a little bit of everything, and that might be why there's more variety.
Thank you very much. Yes. Appreciate it, Grady. Thank you for your time. We will move now, unless there are questions from the group, which I don't see any in chat. No. So I will move on. This is a really interesting topic.
Yeah.
I thought this evaluation of this project was really important. It's an important thing to do. I did have some questions about it. Let's see. One question I had when I looked at your poster was up in the left-hand corner. You say in 2016 the Corps of Engineers completed restoration, and here we had it finishing in 2019. And the data didn't look like 2016 did anything. So what's going on?
So they started looking at the data in 2015, and they finished completing it in 2016. So they did like a 1 year, the barrier complete. So the actual physical barrier was finished in 2016, but they did a year before, so they could have some data at least a little bit before, so they could compare to after. And then they did
Pre-data.
Yes.
Pre-construction.
Yes. And then we kind of left out the during because with a lot of the tests for nutrients and stuff in the TSS, it's stirring up so much stuff of building things. We didn't feel like it was really necessary. So then the afterward, we actually have a mistake was made on our part. This isn't our final version of the chart. Originally, there was a better version with a bar chart with standard error that showed a little bit more, so that's our fault. But with the after 2016, and it goes all the way to 2022, the Army Corps stopped at 2019. So that's when they stopped doing their survey because they thought that they fulfilled what they needed to, which they just did not want to increase the trophic state. They wanted to either keep it the same or decrease it. They just didn't. Yes.
Maintaining trophic.
Yes. Prevent erosion, which they did, and stop trophic state from increasing. So they basically fulfilled it. That's why they stopped reviewing it. And then other people, I think like Dr. Chislock and some of his peers, started to continue also doing more data collection.
Thank you for that. That clarifies things quite a bit. It looks like you got some sort of mixed results here. Do you want to comment on the nature of those results?
Yeah, so 2022 was our outlier, but you can still see a pretty clear trend. Like the pre-construction of the barrier is obviously higher chlorophyll A. And then after the construction, you can see a pretty clear trend that it's a lot lower in trophic state.
Right. I think the earlier question clarifies it a little bit because construction sort of ended here.
Yep.
So between here and here, you're saying that there is a big difference. 22 is the outlier.
Yeah. Construction, like you said, is stirring everything up. So there's less productive. It's less productive. It's going to take a long time for all that to settle out post construction, and then you can see it's increasing back up again.
Do you have an explanation for why this seems to be trending up in the last 2 years?
I think going off of what Liam said, the magnitude of building something like that in the water, especially when Lake Ontario tries to put things into the bay, the tributaries try to push things out of the lakes into the bay. There's so much stuff going in, and then building something that stirs up so much nutrients that was stored in the soil, like phosphorus and stuff, and also the TSS. After, I feel like it takes a couple of years to start to see the trends again, because this is still spinning solids that they went down, but during it was very high. So after running all of my statistical tests, we could take that up because during just crazy amounts of stuff. And then afterwards, 2020, if we were to continue it, I would guarantee there would be a trend, definitely increase in these as well, which seem to be more data.
So that is a long-term trend, not a state.
If we believe that it'll probably stay the same trophic state, but the levels will probably get similar to what they were pre.
Understood. So you get back up to that pre-state.
Yeah, without getting rid of any more shoreline, which is what they basically wanted.
Very good. Very good. Well, thank you for this interesting look at the results of some effort that the Corps of Engineers put in place. And, nicely done.
Thank you. Thank you.
Good work. Thanks, guys.
Problem. Thank you.
We move on to-
Yeah, do you want help trying to figure out how to?
Can we get some of these chairs?
Yeah.
Some chairs.
I just saw the question submitted, came in late. What was the construction? The construction was the restoration of a barrier island in Braddock Bay along Lake Ontario. That was the construction that they were looking at, with kind of a before and after trophic state evaluation. Sorry for missing the question. Hi, so here we are looking at assessing the effects of aeration on the physiochemical properties of Loon Lake. I have an interest in this particular project because in 2021, to August 3rd to be exact, I went diving in Loon Lake with Dr. Bosch and some others to take a look at the area. So I got to see him up close and personal, and I didn't see any difference. I didn't see any effect.
Yeah.
And that's more or less what this study shows. Is that not the case?
That's correct.
You know, of all the papers, of the projects that I've seen so far, this one is really interesting to me because aeration is often suggested as a very simple, cost-effective solution to problems. And yet here we have anecdotal evidence on my behalf, but more important, called quantitative analysis on your part that says, hey, it really doesn't do that much. It's kind of what we're seeing. That's essentially the case?
Yes. So there's a few reasons that our data looks very inconclusive. So
First, we suggest that since the aerators have only been installed for about 2 years, that there hasn't been enough aeration to make a substantial difference. So we suggest that if the study were continued for a few more years, then a pattern may emerge.
You know, let me just say for the Zoom group, I apologize. I believe this is the last, I got these last 3 out of order. So this is the last poster in the file. So if you're trying to follow along, go to the last poster in the file to double check. Sorry about that.
Oh, no problem. And was there another question?
I don't know. I gotta look at my notes. So I actually did read all these. So I just ignored. I wondered if you had any future plans, given that you know, as you just said, it might take longer for the effect to take place, and you know, beyond graduation, it's hard for you to predict, I suppose. But I wondered if there are future plans for studying Loon Lake and aeration in particular. Anybody want to comment?
So we actually weren't the ones who went out and collected the data. I believe it was Chislak's grad students who went out and did that. So that is something that we will have to figure out. If I'm assuming that they have funding for next year. I know at least they do for next year, but we're not sure how much longer they're going to continue this study. But we know that funding at least till next year, of probably till August of next year, is going to continue.
So at least if there's funding, there's a future hope to follow on and see if your predictions or your hypothesis get proven out over a longer period of time. Well, good. Thank you very much. I appreciate it. Really interesting to see, you know, the results sort of summarized this way. And a couple of years ago, I was swimming in the water and it didn't seem like it was doing much. Very interesting. But thank you.
Thank
Well
you.
done. Okay, so there's a couple more here. So again, this is out of order. So this will be number 8 in the file, not, which I guess is probably the same as it would have been if they were in the correct order. Hypoxia, anoxia, and tropic state, Canisius Lake over time. Hypoxia, anoxia. Let's start out by maybe asking somebody to define for us hypoxia and anoxia. I know you did this during your presentation, but I can refresh us with that.
Oh, so this is a microphone? Yeah. Okay, so hypoxia is just is a below average oxygen content for the lake. And anoxia is the more extreme form of this, which just means a lack of oxygen. It is an extremely low amount of oxygen where nothing can survive. Hypoxia can descend into anoxia, and anoxia can recover into hypoxia. So hypoxia is generally where a scant few organisms can survive, and anoxia is almost nothing can.
We see that find my notes. All right, so when I was looking at your poster, I noticed over here on the left, you said Canisius Lake is westernmost thing like this. Eutrophic, yeah. Canisius Lake is eutrophic, and later on, you're investigating whether it really is eutrophic? Okay, so because later on you concluded it isn't. That's exactly what I was a little confused by that, because it seemed like the data showed it's not.
So down. You want to take some?
Sure. Yeah, I mean, we looked at like 9 reports total, and the data that we calculated for trophic state based on those Carlson values, we found it to be mesotrophic. But it has been accepted as eutrophic in the past. So I think more research going into that would be good. And also there's kind of been like a recent decrease that we've observed. So maybe it has been eutrophic and now it's starting to get away from that.
Thank you. I think it's been spoken about at some of the Canisius Lake Association meetings that Canisius Lake is going from It has been going from eutrophic to mesotrophic. I just wanted to confirm that. That would be a kind of a question in a lot of people's minds. We live in.
Our TSI for our collections this year was 42.5, which would put it as distinctly mesotrophic.
TSI means?
TSI is, well, what we use is a Carlson's TSI, and it is a trophic state index. So a Carlson is when we average all of them together. So the average trophic state of the lake is mesotrophic.
Okay, and this is a, excuse me, a numerical summary of qualitative parameters of the lake.
It's a way of putting inherently non-qualitative data into a qualitative state.
Did you want to comment on that?
I was going to say TSI is kind of a way to categorize lakes in terms of like, is it a really productive lake? Is it a more unproductive lake?
Very good, thank you. You know, it's somebody who's lived on Canisius Lake for, well, I've been swimming in Canisius Lake every year of my life. So I have a vested interest in the quality of the water. It's very nice to see a project like you folks have done here, evaluating the quality of the water and helping us understand what are important parameters and things for us to be looking at with water quality. So I think this is really important for us. So thank you.
Running away.
Just took off. We took off with our last poster.
Good thing this thing has wheels, you know.
Yeah, I'm looking for.
How many people are watching?
Oh,
No, nothing.
people.
I'm sorry, Charlie. What did you say?
12 people watching.
12.
Oh, thank you. 12 people, he said.
Thank you, Charlie.
Thank you.
Let me sneak around the other way and catch that last group. Thank you very much.
Thanks, Aaron. Thank you for your everything.
Excuse me. Pardon me.
Reproduction goal, just like the ag maps they're seeing. And then this is did they accomplish both of those? Yeah, 4 of them did. 4 of them. 4 sites accomplished both the recovery or the reproduction goal and the population.
So how small did the populations get in these places? I mean, you talked about how low the numbers got.
They got less than 1% of their original,
Based on range, is that right?
yeah, and then there was only 2 known populations in New York, so they got like extremely small. They were basically almost wiped out completely.
And do you think the cause was more overfishing or was it environmental degradation? Or?
Yeah, there was 2 big causes, which was like excessive harvest, and then like their habitat alterations, which is like the human cause. Like the digging of the trenches and like all the lakes and rivers and everything.
We would let something like that happen to such a big important fish.
And they didn't know how important these fish were. That's why they basically thought it was like a throwaway species. So they would catch them and throw them away. And then they, after like finally killing so many, they realized how important they were. And then they started killing them for their importance, like oil and stuff. And then yeah.
Are they essential to the lake ecosystem in any way, trophic structure?
They help keep the trophic state in balance in the ecosystem. So, you know, because they can be a predator or prey for other fish. And so they keep the balance of the ecosystem. Wow. Which is not normal for fish, it's pretty abnormal, so that's definitely not helping their numbers maintain.
Very slow population.
Yeah, it's from our, so this is pre-stocking, so they stocked all the locations with sturgeon, with adults, juveniles, egg mass. So this is before they stock them, and you can see how much.
Yeah. Do they eat round gobies? They look like a bottom feeder.
Yeah, they are vegetable feeders, so I can imagine they eat gobies here. So lots to learn about this. So yeah, you can see the past recovery.
Those numbers are just actual 6,465 fish.
And you know, these are just estimates. Were calculated using different forms, like the mark recapture and how they put it online too, like which formulas they use for different as well, but that was too much to get into.
Right.
Yeah, the DEC had some really good figures to also show, because they brought it all the way up to 2024 for the future recovery type shifts. Which we'll look at. They got one on.
Yeah, I see the different methodologies.
Yeah.
Still not a lot.
Yeah.
You make Western Lake Ontario, but all the other one, Lake Champlain is 92.
Yeah.
That's a big lake.
Yeah, so I assume this is like one population, like this is spawning stuff or something, you know.
Well, they're not a pretty.
They look like dinosaurs, yeah.
Yeah, they are.
The oldest supposed to be really fish to dinosaurs.
Is that right? Nice, thank you.
Yeah, you're welcome, thank you.
Maybe you covered this, but can you tell me a little bit more about mark recapture analysis and those other techniques?
Yeah, so mark recapture is basically when you go to a site and like the location you're going to study, they take like 50 fish, 50 sturgeon from the lake from either electrofishing or catching them, however. They'll mark them by either clipping their tail or like drawing on nail polish or something like that on them so they can identify them later. And then they'll go back like 2-3 weeks or months later or multiple times and see how many of those marked fish they recapture. So that's why it's called mark recapture method. Yeah, so that helps them keep track of how many new fish they're getting, how many old fish they've seen again. That kind of helps keep an eye on their actual population status.
So it then becomes kind of a statistical game.
Absolutely.
Okay, got it. Very good. Thank you. You mentioned in one of your discussions, talked about habitat improvement, monitoring, and research. What areas of research? And anybody else want to comment? What areas of research would you think would be important for
expand on?
Expanding in which direction?
Like for future research?
Well, that's what you're suggesting, right?
So more like spawning sites. That's what we yeah, because we have a lack of knowledge of where they spawn, the locations and demographics for many of the populations. Such like field surveys, assessing population statuses and like the habitat improvement are key to like the rehabilitation and enhancement of these lake sturgeon populations. So we need more knowledge on where they spawn, what time they spawn.
So where and when they spawn will help us create conditions that would enable that. Yeah. That makes sense. That's good. I was also, what do you mean here by feasibility limited by egg takes? What's egg take?
That was so feasible, like limited, because artificial reefs are basically destroying the habitat that's already in place to put a like sort of fake habitat. So it's not really like feasible to do. And then for egg takes, I found that when they did do the egg takes, they would find like there to be 100% mortality of the eggs that were taken from the sites and habitats.
I'm confused. An egg take is taking eggs from a breeding site and
I'm pretty sure I can't remember the whole thing, but they saw that there was 100% mortality in that method.
so whatever an egg take is, it's not successful. Okay, I got it. So they're trying to maybe take the eggs and put them in conditions that would be more survivable, and they turned out not to be. Interesting. Do you have any other questions? Those are my questions. I really appreciate that you were taking a look at some real life conditions, not associated with Geneseo Lake, which I'm most familiar with, but with a very important fish in the Great Lakes. So thank you for that. I appreciate it. I was always struck by the difference between the Geneseo focus of it's like biology and food web kind of things.
Yeah.
And you folks are much more interested in the physical environment.
Yeah, they kind of throw us right into it.
Yeah.
Into the physical part of it.
Very interesting. You can really see the difference in focus of the courses.
Yeah.
Thank you very much for your time.
Thank you so much.
Nice to meet you too. Well done. So that's it for me from the WEC. Let's see, I don't see any other questions.
It's fine to say, good to see you.
Good to see you.
Have fun in Antarctica.
We'll do. I'm looking forward to it. Please get up in here. I can't hear you.
No questions.
What's that, Charlie?
No questions. There are none.
Hi, Charlie.
Hey, Zen. Ray.
Sorry, there's enough noise here that it's very difficult to hear.
Yeah, turn it over to Ray.
Here's Ray, right here.
Hi, Charlie.
Hey. Certificates.
You can't, it's really hard to hear you, Charlie. I'm sorry.
Certificates.
I could turn the overhead microphone.
Yeah.
Certificates. Thank you, Charlie.
Yeah.
I'll remind you. So have you been certificates? Certificates? I actually Mike, he's passed them over. The kids are starting to leave. Because the students have needs to leave early, he's giving them to the professors who are passing them out. Thank you for the reminder, Charlie. All right, I'm going to close out here. I'm going to leave the meeting.